everyone, and welcome to the first episode we are shooting for Colloquy. So, as many of you have already seen on our Facebook page, now Colloquy is part of an initiative which I have started called Emulsion. Because in the recent times, as we are seeing that science is having so much misinformation being thrown around with the wake of coronavirus, we really don't know exactly how science is working and what are the intricate details of it. And a lot of it is in scientific journals. So Emulsion was started as an idea to get science to a larger group of people at a technical level, but still understandable by a large population. So as part of this, I thought of starting Colloquy, a regular discussion we'll be having with people, scientists, students all over the world, and talking to them about their field of expertise, about questions we don't know, getting into the technical details, but still making it fun and interesting for all of us. For the first episode, we have Jash Mota. He is the co-founder of Centauri Robotics. And say hi, Jash.
Hi.
Yeah, and he's a very good friend of mine. We have known each other for more than a decade now, and I thought I should be starting it with him because he is extremely good at robotics and I'm not. And my field and my, I won't say expertise, but it's my area is more to the biochemistry and biophysics. So I thought that we have never really had the chance to discuss and explain our areas of work to each other, and why not we do it so that everybody can understand and we can find more about each other's work too. So that was the basic idea how we started, and that's how the first episode we are trying to unscript in Colloquy. So with that, Josh, would you like to have some kind of introductory words?
Sure. So, I'm really happy to do this. In times like these, where coronavirus pandemic is just spreading all over the world, you really wish you knew more about the underlying virus and how this thing works and how it spreads around. So you know the underlying mechanics of it. So it's going to be really good to talk about it, to get to know more about it, probably learn how to reduce the spread and how we can, uh, just be more aware about it in the future as well.
Yeah, that's great. Thank you so much, Josh, for that motivating words. It's the first time we are doing this, so let's see how it goes. But
Sure.
yeah, so first of all, how are you? Are you doing well with the entire Corona thing? How is it back in India?
Well, the cases are rising. I think it's somewhere around 25,000 today. Um, fortunately, my family, everyone in my family is safe, so that's good. The lockdown is definitely working, and so the only thing I know about coronavirus or the entire thing is that it's a virus, and it spreads through when people sneeze, with the infected people sneeze or they cough or even they breathe. And but I've seen all the sort of absurd things that people are doing all over the world. Like it goes to very extremes of the spectrum, where some people are just very careless about it, and they say it's probably just nothing. And there's the other end of the spectrum, which thinks that maybe drinking bleach can keep them safe.
Yeah. So yeah, that's actually, yeah, it's been quite recently,
And
especially with the President Donald Trump's comments. It's quite going around.
and what really surprises and kind of frustrates me is I don't really understand which end of spectrum is the less scientific one. I mean, uh, drinking bleach definitely sounds absurd, but I do not have a good measure to say if it will work or not. Definitely, it's going to be a very bad decision for your body. But yeah, so those things just make me feel, damn, I should know more about uh, how the entire the concepts of virus and proteins work. And if proteins are in virus, why aren't they good? And so just tell me, does drinking bleach help in keeping you safe from coronavirus?
Yeah, so the simple, shortest answer is no, no, no. Drinking bleach
Thank God.
doesn't help at all. Because you have to understand the basic point. It's called bleach for a reason. It's going to strip you out of something. We use the word bleach in something, very strong sense. It's going to strip you out of all colors or something. So the disinfectant which we are using at our homes is very, very strong. We have sometimes chlorine and other kind of chemicals, especially if you, I don't know if you have, we smell chlorine in the swimming pools, but if you just have chlorine gas in front of you, it's quite pungent. You can't even stand the smell of it even for a second. So these are extremely harmful. It's chemicals. It's straight up laboratory chemicals, and they are in no way to be consumed. But it's like the same logic. Maybe if you take a virus and put a bleach on it, and maybe it will die. But that doesn't mean you will drink the bleach and it's going to kill the virus. It's almost like saying that you have iron deficiency of anemia and you start chewing on an iron rod. That doesn't work. So we really have to understand that that's not scientific. Science, and especially human body, is far more complex. We have a system, and more or less it will just burn your stomach and just go out as waste. That's all is going to happen. So you got a good question, because exactly how this virus exactly is working, and why is it so dangerous itself, because it's such a widespread. And you spoke about, yeah, that's true, that the coronavirus itself, on the outside, it has a protein layer. And it's one of the proteins which is actually from the scientific community, which is very important in coronavirus, is the spike protein. So that's a very small kind of protein. It's spike. It's almost like comes out of a ball. So this is a virus, and it's like a spike protein. And that spike protein actually goes and attaches to some cells in your lungs when you breathe, or when you breathe the virus, or just inhale it. It goes and attaches to the cells of the lungs. So what happens? The cells of the lungs especially are depend. What they want is to take in more oxygen. That's what they are there in the lungs for the respiration. Now, if the virus attaches to it, this through the spike protein, the oxygen cannot bind to it. The oxygen cannot attach to those cells. So what you're going to be facing is the lack of oxygen. That is why you have the respiratory problems in coronavirus, breathlessness, and followed by this pneumonia in the later stages. Because this virus is not allowing the oxygen to bind to the cells in your respiratory cells, it's for that matter. So that is exactly the problem with the spike protein. And that's actually the more damaging factor in coronavirus.
Makes a lot more sense now that they are kind of jamming up your respiratory system.
That's right. That's right.
So talking about the structure of the virus, it's kind of it's spherical and has like radial spikes radially outwards. Yeah. So the images we see in the every different kind of social media story is kind of correct.
Yeah,
kind of accurate.
Actually correct. That's true. So one of the first images, because there is a Chinese group, I forgot the name. It's from a research university in Wuhan itself, and the lady professor, she has been working with coronaviruses for more than a decade now, since 2000s. And they once this outbreak happened back in China, the first things they did is to find the structure of the virus. So that is very important in any kind of cure, that if we are trying to deal with an organism, we at least need to know who our enemy is. How does the enemy look? So they brought out the first structures back and published in the journals, which sped up the process of finding out, okay, so what we can do, what exactly it is causing. And they found that the structure is very similar to the SARS virus outbreak, which happened back in 2003. And it's similar, but it is still, there are some differences in the sequence and structure. But yeah, that they identified that, yeah, this is the spike protein which is actually causing it. So on the social media, the structures you see spherical with spikes coming out, that's accurate. That's accurate.
Okay. So I didn't really know that structure of a virus was so important. So can you just tell us more about, so how do you, in brief, if you can tell us about how do you find the structure? Because they are very small organisms, they are microorganisms. So how do you find the structure of the virus?
So what exactly we do is we try to get crystal images. Now, what are crystal images? It's like, it's very much in the name that we try to make like a crystal out of the virus itself so that it can have like a solid structure, because if it is solid, it's easier to image. So it's like, for example, like if you take like a salt, or if you take like a sugar. Now, sugar has, for example, carbon, hydrogen, and oxygen in each part of sugar, and they are not just random. They're arranged like in a proper cubical structure kind of thing. So even with the something of a similar idea, we try to make crystals of these kind of viruses, try to make them like kind of stationary, and then we image them by passing the X-rays. So we bounce off X-rays, and we use X-ray. We use X-ray for our X-ray checkup in medical. We use X-ray for finding the structures of these proteins also, and this virus. So through that, we can get multiple images, and we can find and we can club them together and get a 3D image. But yeah, we don't get like a colorful image like what we see in the news media. No, we don't see it red, blue, and things like that. But that's just for identifying different parts. But that is the X-ray crystal structure is what we use to get the structure of the virus and the protein on it.
That's pretty interesting.
Mm-hmm.
So what all are the constituents of the virus? Like, what are the different parts? And how is the spikes made up of, and what's inside inside that spherical structure?
So, for example, just in brief, if I have to explain that the virus, so first of all, in the scientific community also, we do not really classify virus as living or non-living. It's somewhere at the border of it. That's also a very interesting thing. And because virus is technically not living outside a cell, it's only when it infects a cell, it reproduces. So that is why the definition of living and non-living is a little confused in the virus. But the virus, yeah.
So it's like it's not living until it has it is inside a body or some kind of environment where it can reproduce.
That's what we can we can say, that it has to find something to reproduce, something to grow on. So that is exactly what viruses do. But for the structural part that you asked, so virus, it has like a oil layer outside. It is it is an oil layer, let's say like a fat layer, and from the fat layer we have those proteins coming out. Okay, so that's why these proteins, we call them as glycoproteins. Okay, we'll talk about it later on. But it is like a fat layer, and there are proteins coming out. Now, why are we saying that washing hands is good? Because, just imagine if you're cooking something and if your hand is oily, when you just wash with water, it doesn't come out, because water doesn't mix with oil. But when you when you wash it with soap, you clean your hands. Because what the soap does is like surrounds this oil molecule and mixes it with water and then washes it off. So that is exactly what is happening with washing hands. If you wash hands, the soap is going to cover the virus molecule, and it's going to break apart the oil and the fat layer, and in a sense killing the virus itself. So that's also, we talk about why hand washing is so important for such kind of viruses. And from the fat layer, we have these spiky proteins. Now, you mentioned about it, because for proteins, we don't really imagine them to be something harmful. We always imagine from the diet point of view. But these proteins are, I won't say, it's very difficult, but it's this, all proteins have similar structure, like in the sense like the basic structure is the same, but the different atoms actually and the different shapes make it different. But it's one of the kind of proteins, it's there on the top of the virus, which is infecting the cells.
I see. So so there's a layer of fat which covers the entire sphere.
That's right.
And there's spikes, which basically is protein. And inside there is?
So inside it's just a viral. You have the genome, which is the RNA or the RNA of the virus. So are you familiar
Oh,
with RNA?
not really. So what's an RNA?
Okay. So all right. So let me just first tell you, like as you asked, that inside, there's a fat layer and there's a protein, but inside you have an RNA. It is this RNA which is actually, so what happens when a virus, just imagine it to be like, for example, if this is your cell, so the virus comes and attaches to it. Now your protein and all is okay, attached right on the top. But what is the virus doing inside the cell? It is it is going to inject something called as a RNA. Now that multiplies and destroys the cell and things like that. Similar mechanism in HIV, similar mechanism in most viral diseases. But what is RNA exactly? It's called as a ribonucleic acid. And uh, uh, now you would think it's like an acid like any other, like a hydrochloric acid or something like that, but not really. So it might be better if I show you a structure. So maybe just hold on. I have some structures of DNA and RNA prepared. So maybe I can just share it with you and try to explain.
That's great.
Yeah. Okay.
Sure.
So I'll do the screen share thing. All right. Uh, yeah. So yeah. Uh, eh, hmm. So for example, okay, because to understand RNA, it's easier to understand what a DNA is exactly, because DNA is something, have you come across the word DNA?
Yeah.
Okay, so what is your idea of DNA? Like, what do you think it is?
So I know it contains information about individual,
Mhm.
and it's a helical structure.
That's right. So,
But
mhm.
yeah, I do not really know what are the constituents and how it actually works, and what's the how does it what's the size and how is it relative to the other cells and stuff like that.
Right. So that's a very good understanding, and I think it's quite it's a more than average what I had expected from a general population, but that's true. So DNA is like it's almost, let's say, it's the memory, it's the USB drive of the human body. It is what is important, what is needed to pass on the information. So from parents to children, from their children to from one generation to other generation, when the information that is being passed in something called as DNA. Now DNA is called as a deoxyribonucleic acid. Okay.
That's a mouthful.
Mm. Yeah. uh, Okay. So we have, it's called as a deoxyribonucleic acid, and as you said that it looks like this. It's a helical structure. So it's almost like a one giant snake with these two strings attached to each other. But it's not that simple, actually. I mean, this is the very simple picture which we see. But in DNA, you have these, as you can see here, adenine, thymine, guanine. Because you don't, the names don't matter. It's A, T, G, and C, as you can see here. Okay.
That makes it simpler.
Yeah, it's very simple. Don't remember the names. Even I confuse the structures a lot of times. It's fine. So
That's really
yeah. So so like, so A and T always bind to each other, and C and G
okay.
always bind to each other. That's a rule. That's just the way it is. So the number of A will be equal to number of T, and the number of C will be equal to number of G.
Okay.
It's always like that. Okay, so that is what you see, is this bars which you see, these rods, yellow,
Yeah.
white and green. So they are these structures. So it's better clear in this on your right side. I think you can see my mouse.
Yeah.
Yeah. So for example, you have A and T. This is, and then C and G. So you might be noticing that A and T are there is like two pink lines here.
Yes.
So that's, let's say they're like bonds. So like, how do A and T stay attached to each other? Okay. So for example, okay, like if I meet you in person and I have to stay attached to you, So what will I do? I'll probably hold your hand. So that
Yeah.
holding your hand, let's say that's a bond.
All right.
Okay. So A and T make 2 bonds, and C and
Okay.
G make 3 bonds.
Okay.
Yeah, like back in India in high school, we used to remember like Karishma Kapoor and Govinda used to have more films than Amitabh and Tina Munim. So you have more bonds in ANG and lesser bonds in ANT. Well, that's just pop culture reference, but yeah. So we have these kind of bondings happening. But what is this blue and yellow? You might be wondering. So
Yeah.
this blue and yellow is what you see as a ribbon here, this backbone, which is called as a sugar phosphate backbone.
Oh, I see.
So it is actually just your normal sugar. I'm not going to be dealing with the structure because it's very much into organic chemistry. It will be a completely different lesson altogether. But what you have actually in this ribbon is sugar and phosphorus.
Okay.
So, and it is just that sugar and phosphorus holding these kind of A, T, G, C molecules in a very, 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 very long sequence. That is exactly what DNA is.
Oh,
Okay. So,
that makes it bit.
so because I mean, you must be wondering what is the size, because I'm telling it's very long, but
Yeah. So how long is it?
so like see, between the two ribbon, what you see, the distance is 2 nanometers.
Okay.
So now nanometers, like just to imagine it, for example, um, uh, the length of, let's say, the beard which grows in an average human per second is 5 nanometers.
Okay. So that's very small.
That's extremely small. That's extremely small. So it's like if you divide 1 meter into 1 billion pieces, each
Mhm.
piece is 1 nanometer. So the distance between these two ribbons is about 2 nanometers. So it's not very wide. But I will tell you something interesting later on. Like, because uh, let's say, because the DNA, because our body, every living body is made up of cells, right?
Yeah.
So these cells are everything, like every single skin and everything is made up of cells. You might be hearing these especially in the beauty commercials, when they say that your skin cells need to breathe and open up.
Yeah.
It's yeah, it's the same thing. It's the cells. So inside the cell there is something called as a nucleus, which is like a heart of the cell.
Okay.
So think of the cell as a human body itself, and the nucleus is the heart.
Okay.
Okay. So inside this nucleus, you have this kind of structure, as you can see my mouse going around, this
Hmm.
kind of uh, like a tongue type structure called chromosome.
Okay.
Okay. So human beings actually have 23 pairs of this kind of structure, this chromosome. Your like when we say male, female, X chromosome, Y chromosome, this is how the chromosome looks like.
I see.
But if you see, like, if you might be like what exactly is this made up of? So this chromosome is made up of a structure, this this kind of coil barb like structure you can see.
Yeah.
It is made up of this. Now if you zoom more into it, you will see this kind of structure, this heavily dense circular disc, almost looks like a candy with some ropes around it.
Yeah.
So, and each of this rope which you see is actually the DNA.
Okay.
So inside the cell, you go inside the nucleus, you go inside this chromosome, then inside this coil structure, this small little rope you see, that is DNA.
That's amazing. That's like a lot of beautiful chaos happening at a very small scale.
It is, and it's so, you'll be very interested to know that. Okay, let me ask you. Guess the total length of the DNA in this one small cell. The total length.
I'm guessing 100 nanometers.
100 nanometers. Well, you'll be surprised. It's actually 2 meters.
2 meters.
2 meters in every single cell. because it is so densely packed that of course, we can't see it, but the packaging is so beautiful that 2 meter length of material could be shrunk into something so small.
That just seems impossible. I mean, oh, you just blew away my mind.
Well, that's exactly. I mean, our human cells are extremely interesting. But since coming back to your question, because this is what I wanted to give you an understanding of the DNA itself, so that it's easy for me to tell you about RNA.
Okay.
So for example, like let's see this structure. So as I told you, DNA has these two kind of ribbons, right?
Yeah.
Now RNA is just only single ribbon.
Okay.
Okay, it is not splitting the DNA into half and half. No, it is a different structure completely, but it is like a single ribbon. Now, if you want to ask like what exactly is the difference between a DNA and a RNA, because like see in DNA we talked about these 4 bases called A, G, C, and T.
Okay.
In RNA, instead of T, we have U, something called uracil. That's the only difference.
Okay.
Okay, and everything else is the same. And even between T and U, if you just look closely, just see this structure of T here and this structure of U here. The only difference is like 1 carbon and 3 hydrogen. That's it.
That's a very small difference. So is it, how big of a difference does it create?
Everything. It changes everything. And just to tell you like how exactly things are similar, I'm telling you like between a human being and an orangutan, 97% of a DNA is the same. It's only the 3% which makes a difference.
Are you kidding me right now?
That's exactly the thing.
That is impossible.
That's actually the 3% which is actually making us so different. So all of these questions about, I mean, we are so different from animals. We are not. Majority of our DNA is exactly the same.
That, oh my God, that's definitely, that just blows up my mind.
That's yeah, actually I was also very surprised when I came to know back in high school. I was like, oh, so we are almost like orangutans. Interesting. So this is what RNA is. Okay, so the RNA is like this one ribbon, but you might be you might be getting a feeling from the structure that wait, this kind of these rods don't seem like bound like the way you can see in DNA. DNA looks like a closed structure. RNA looks like more open.
Yeah.
So RNA, yes, it is more open, and that is also what makes it more reactive.
Yeah, that's, I mean, so I think it's more hungry for making those bonds. So it is just looking for anything which will help it make bonds.
That's right. It's actually like that. It's very reactive. It's very hungry. And this is the thing which is there inside the virus, inside your coronavirus, this RNA. And the problem is that, because DNA is, I mean, it's not permanent. The DNA is always like, it's duplicated, triplicated, it is like double, tripled every time. The cells are constantly making new and new DNA. Okay. So, but when you make a DNA, the amount of mistakes the cell makes is extremely very low. Like, if it makes 1 billion of these bases, C, G, A, T, 1 billion bases, it will make one mistake, probably.
Oh, that's a very small probability to make a mistake.
It's very precise. But RNA does not do that. RNA makes a lot more mistakes, actually. And the mistakes in the RNA makes it mutate much faster.
Okay.
Because it's changing all the time, because it's making mistakes constantly, it changes very fast. And that is exactly why, when you hear that, why are we finding it so difficult to find a cure for a virus or coronavirus? Because RNA is always changing. So you make something to attack this RNA and it has already changed.
Oh, that makes sense. What makes it so difficult to contain the virus?
Exactly. And we cannot predict this change. we cannot predict which part it will make a mistake.
I see.
So since we
So
cannot predict that, we cannot predict mutation, and thus we cannot predict evolution.
that's right. So the mutation, the generation of the RNA is it's not deterministic. So we don't know what it's going to do next. So am I right?
Yeah, so we cannot really know what exactly it's going to change. For example, okay, maybe I'll just tell you, like, first of all, how do they make a new DNA and an RNA? Maybe that might just clear up your things about how exactly are they making a mistake or not making a mistake.
Yeah, sure.
So for example, let's see, like, this is, um, uh, because since you asked, you might be interested to know that actually proteins are made from RNA and RNA is made from DNA.
Okay.
So that is actually, it's something very central to cell biology, to every single living organism, that DNA makes RNA and RNA makes protein. It's called central dogma. It's so important. Every single thing behaves like that. So so I will probably just give, because since we are talking about why RNA and how RNA makes these mistakes, and how it relates to the spike protein or something like that. So maybe it might be better if I just tell you how DNA and RNA and protein are made from each other. So, as we saw it in the previous structure, like, as you can see, this is the two ribbon kind of DNA, right? So so what happens, like, for example, let's see. Okay, like, I'll give an example. Maybe I'll just stop the screen share for some time. Okay. So, like, let's say, like, these are the two little ribbons of the DNA which you see. And how are they arranged is, like, they are just helical structure that is coiled against each other. So yeah, I don't think this ribbon is, this is a ribbon I use for actually throwing away my trash every Thursday, but fine. So it looks like something like this. It's coiled, and it's like two different ribbons are bound against each other, they're wrapped with each other. So what happens is that if you want to make a new DNA, you need a template. How do you make a new DNA? You just can't make a random DNA, right? Because as we discussed, it stores our information. And if it stores your information, it can't be random. It has to be exactly the way it was before.
Right.
So what it does is, there are a lot of different proteins which actually work in making a new DNA. So what they do is they open up the structure. They open up the ribbon into two separate ribbons. Okay. Now, as I told you that the A is always bond with T, and C is always bond with G. So you have a rule. So that's the system knows, okay, if it's an A, I will make a T. If it's a C, I will make a G. Yeah. So there is this protein called, it's a different protein. Its name is called DNA polymerase, but let's skip that. It's a protein. It actually, what it does is it bounds to the DNA, let's say here, and then it just sees what kind of bases are here, like A, C, G, T, G, C, A, and all. And then it just keeps on adding the complementary bases, T, T, G, like that. So it just like, so what happens is, like, at this side, the ribbon keeps on opening up more and more bases start to add and make a new DNA.
All right.
So what happens?
So there's these two strands.
Yes.
And you kind of read through each step.
Yes.
And then you find out what molecule is over there. And then you add the complementary molecule. And that's how basically from a single DNA you get to.
That's right. So at the end, what will happen is, like, you'll have one individual and one old one and one new guy here, directly parallel to it.
All right.
Okay. So then you will, from like 2 ribbons of DNA, now you get 4. But here is an interesting part. The protein which actually makes this DNA, the new DNA, it actually works only in one direction. So,
What does it mean?
so for example, the DNA 2 ribbons which you see, okay, they look parallel, but they're not. They're actually anti-parallel.
Okay.
Okay. So, like for example, if the DNA, the protein which makes the new DNA goes in this direction, it cannot go in this direction.
All right.
Okay. So
basically, it opens up only from one side.
So what happens is, well, when it's opening up, the 2 sides are opening up, right? So one side, it will be very normal, continuous, it's making. But the other side, what it does is, it makes it in the opposite direction, little by little. A little, then leaves it, again joins, little, again leaves it, again joins. So what it leaves is like a small fragments, and then they join together to make a complete DNA. But in short, because I mean, I'm not going to get into details. It's like from one DNA, I'll again, like, yeah, just show you here. So as you can see, from one DNA, you get these kind of opposite to DNA, and you get a new DNA in short.
Yeah.
Okay. But once this DNA, that's how DNA actually replicates. It's doubles and triples kind of thing. Now, if you have to make a RNA from DNA, so what you actually do is something like this. So for example, I made a very simple structure for you because I thought that this might be coming up in a conversation. So as you can see, this pink guy is the DNA. Okay. And there is a protein. It's called RNA polymerase, but you don't have to read it. There's a protein. Okay, there's a protein which comes to the DNA and attaches. And again, because as you remember, the RNA has the same basis except for the T and the U.
Yeah.
So it can also follow the same rule. The A with, instead of T, with A with U and C with G.
Yeah.
So what it does is it also opens up the 2 DNA ribbons and uses one of the ribbon and makes a complementary, like again, the same, like a complementary molecules are being added. And that is how it only does it once, because as you remember, just one ribbon.
Yeah.
Okay, it just does it once, and here you get as something as RNA.
Okay.
It's called mRNA because it's called messenger RNA, but it's fine, but it's an RNA. So what?
Just
yeah, go on.
Abstracting away from that.
Yeah.
So RNA basically hampers the normal DNA reproduction process, if we call it like that.
No, we cannot actually, because they both actually work together. It makes more and more DNA also. It makes more and more RNA also.
Okay. So RNA is present in every cell.
Yes, it is.
So so that RNA structure,
yes,
is obviously going to be different from the virus.
Yes, obviously, yes.
Okay, and so RNA and DNA,
yes,
bonding together,
yes,
is not definitely a bad thing. I mean, it's in fact a normal thing, as you are explaining.
Yeah, of course, because RNA is made from DNA. So of course, there is some binding between RNA and DNA happening.
So it actually depends on what RNA is bonding with DNA, which can make it normal or can make it dangerous.
Well, that's true. That's exactly the truth. That's exactly the fact. And it's same for every single living organism. For a single small microorganism, bacteria like E. coli also, they have similar problems. So they do it exactly in the same way. That's true. It only depends on what kind of RNA.
So I'm trying to understand a bit now.
Yes.
So when this virus attacks these cells in your lungs, as you explained before,
yeah,
what it is doing, I assume, is the RNA from the virus
yes,
intervenes in the process of this bonding with the individual strands of DNA.
Yes.
And then creates kind of, let's say, a rogue DNA.
Okay, yeah, it's very simplistically, yeah, we can say that it creates like a rogue DNA.
And then that rogue population increases and affects your body.
Yeah. So there is something which I must add here right now.
Okay.
Viruses have a special ability to make DNA from RNA.
Okay.
Okay, what I said earlier was that DNA to RNA to protein, that's the process, right?
Yeah.
But virus can make RNA to DNA also.
That seems a bit contradictory because RNAs have this component U, which is different from T. So
That's
how does it actually do that?
So there are some proteins which are there, which are always helping in this process. And though it is reading, it is going to read something complementary, right? So,
Yeah.
for example, if there is an A on the RNA, you might think, oh, it should get a U in the complementary. But what it is attracting will probably be a T from the DNA sequence. So there are some proteins which specially work with the virus RNA and try to make this DNA. It's actually the name is called reverse transcriptase, but I'm not getting into the names and stuff. But it's called reverse for a reason, because it can go back, it can make a DNA out of RNA. So what
That,
happens? Yeah, go on.
so you can basically reverse the process, go from RNA to a DNA.
So why it actually why inform this is because what happens is, if you make a DNA from a RNA, and now you have a normal human cell or whatever cell you have, okay, if
Yeah.
there is DNA, the normal cell will make RNA and proteins out of it.
Yeah.
So what you're actually making is like you're supplying some initial materials and making a factory to reproduce.
Yeah.
So you have your RNA inside, and through that you're making a DNA and giving it to the cell. The cell just takes the DNA and makes its own RNA proteins, divides, reproduces, sends it out, infects others, and does it like that. So that is like a factory. That cell becomes like a factory for the virus to produce more and more viruses.
Okay.
So that is why it uses the DNA to kind of trick the cell to follow its DNA, RNA, protein process and infect it further.
So first it attacks the DNA, which then attacks the cell, and then then the cell attacks the other cells.
Yeah, the cells again. I mean, the cells can also reproduce. The cells also release these proteins outside, and also a lot of other things keep on happening. And then, so the virus uses the cell, exploits, almost like makes a slave out of the cell for its own benefit.
Okay, that's interesting.
That's exactly. I mean, that's evolutionary reasons. Of course, I mean, virus needs to survive. Otherwise, if it was too simple, I mean, normal cells could just kill away the virus.
Yeah,
they have developed their own defense mechanisms also.
But it will definitely, it will be more cool if we don't have any cough or cold in this world.
Right. I mean, that's the reason we don't have a vaccine for it. Like, it's common cold. It's so common that we
exactly.
don't have...
It's so common, we named it common cold. We just have to live with it.
Yeah, that's right. I mean, that's the thing. I mean, viruses are very complex. There are millions we haven't discovered yet, and there are always new things to keep on discovering. So just returning back, because we are talking about the DNA, RNA, protein process. So as I told you, from the DNA, we can make this RNA, and this RNA can attach, uses some other proteins called as ribosomes. I have shown you the picture. I'll go back to that picture so that it's easy for you to image. So for example, you see the DNA is written here inside the nucleus. I said the heart of the cell.
Yeah.
So inside there, you make the RNA. But once the RNA is made, it comes out in this area, in this light blue, big, large playground kind of area. Okay.
Yeah.
And this, kind of like wiggly woggly brick-like structure, orange structure you're seeing, it's called as the endoplasmic reticulum. Doesn't matter, but this wiggly woggly structure, that is where you actually make the proteins using this guy called ribosome.
Oh, so it's that tiny specks in
Yes, like the
that blue cytoplasm.
that's right. It's called the cytoplasm, but these small things are almost like the seeds of a watermelon. But
Okay.
what we do is here, see, you can see the ribosomes here, written here.
Mhm.
These ribosomes bind to this RNA and actually make like a long chain, which is called as a peptide chain. It is one long chain. It's a chain. Okay.
Okay.
This chain then folds on itself, and that is how we get a protein.
But isn't ribosome itself protein? So
That's true.
what is the resulting protein? What is
So
it called?
that's
And what does it do?
that's a very interesting question. Yes. So it depends very much on the DNA sequence. So whatever,
Okay.
so because I said A, G, C, T, their bases are there. They're information. So A, G,
Right.
C, T is an information which is different from A, C, G, T.
All right. So uh, just a minute.
Yes.
I was thinking that it's kind of an alternating sequence. So is it,
Hmm.
or it can be a random sequence?
It's random. That's exactly the thing. It's absolutely random.
So what does it depend on?
It does not, well, what exactly the sequence depend on, that's evolutionary. That depends on what kind
So
of,
it's completely random.
it's complete, well, just to look at it, it's random. But why exactly each of that molecule A, G, C, T is present, that has a reason which we do not know, but of course nature has a reason for it.
All right. So we do not. So it it might be deterministic, but we do not know yet.
So like, just to give you example how big the sequence is, I said like it's 2 meter long, right?
Yeah.
So, just in one single DNA, just imagine if you have, for example, if you have 3,000 books, and each
Mhm.
book has 1,000 pages, and each
Yeah.
page has 1,000 letters. Okay.
Oh,
Okay. Now the total number of letters in the 3,000 books which you have,
mhm.
that number of letters is present in the single DNA, and each letter is A, G, or C, or T. Each letter.
Oh, that's a lot.
That's a lot. So if you just imagine if each letter can have four choices, A, G, C, or T,
Mhm.
you have so many choices and to create such random sequences.
Yeah, there's a lot of permutations.
It's not a permutations, right? I mean, it's nearly impossible uh, to to calculate the number of possibilities are possible. So depending on what kind of arrangement is there, it will make
But
like a
this is
complicated.
interesting. This is an interesting combinatoric. So sorry to interrupt, but
No, no, sure, sure.
uh, so if we have a DNA sequence
Yeah.
inside a person's body, is that sequence the same throughout all the cells in that human body or any kind of creature, for example?
Uh,
Or
in the same human body, yes.
okay,
The DNA sequence will be the same.
that's interesting.
That will exactly be the same. That has to be the same.
All right, so um, I think we have about 1 billion uh, different spots,
3 billion.
and then sorry.
3 billion. Like I said, like 3,000 books, so 3 billion.
Okay, so we have 3 billion, and the permutations of 3 billion spots. So
Yes.
we can have that many different unique kind of humans, or not just humans, actually the entire.
Living.
Uh, that's a lot.
That's a lot. So like 4 times 4 times 4, so 4 raised to 3 billion chances are there to
Yeah,
make the entire thing.
and it might seem that 3 billion spots is not a lot, but when you add the permutations of 3 billion spots, it's a very gigantic number.
It is a gigantic number. And the thing is, the 3 billion, it's for a human cell. Different organisms don't have, everybody doesn't have 3 billion. Somebody have like a 1 billion.
Oh,
The length is different from organism to organism also. So that, so just imagine in every single organism, you can again further make those permutations.
this is pretty interesting.
So that's it's almost infinite at a human level, because since we cannot count,
Yeah,
almost infinite.
that's true.
So this sequence determines what exactly, because the rule is set, A, G, C, T, or U. The rule is set. Depending on that, you get the RNA, and depending on the same rule, you get the protein.
All right.
Okay, so it's that. So the living system knows how precise it is, but we still have to find out how does it work. But to add on to that, because like for example, I told you this kind of this peptide chain is folding. Okay,
And it depends on the DNA sequence.
it depends. Well, we assume to be depending on the DNA sequence, but this folding process is precise. It's not a random fold. It is
Oh, that's
exact. So you
oh,
take this folded structure,
you are
fold it.
you are literally blowing my mind every second.
See, it's fun, right? Like, just imagine you have so many options, so many permutations, and you get this one structure, a folded protein structure, and you fold it in any way, but it will still return to one single structure, and we do not know how does it do that.
That's pretty amazing. So evolution has been doing some pretty heavyweight stuff
Oh, they have been
for
working.
these years.
There is no corona time, corona quarantine for evolution. They have been working 24 hours.
That's right.
So the
All
protein.
right. So yeah, we have this protein fold.
Yes.
So what's the end result or after the fold?
So this is the protein. Okay, this is exactly what we call as a protein. Every single thing.
Okay.
You say protein. This is this protein. Now, because every protein has, I gave it by a different shape here. Now, as you can see, the shapes are unique, and they're unique for a reason. They're almost like, Lego blocks. Like, one protein might actually go and fit into something else, like a proper lock and key structure.
Okay.
So if it fits, it acts like a signal for some processes to happen.
That's
So that is how it determines. For example, maybe when you, like for diabetes people, we say that the insulin production is not enough. So when you have too much sugar, the insulin level should go up. And when you have less, it should come down. Who determines this process? Who realizes that? How does the body know that how much insulin should I produce? That is all dependent on these proteins. These proteins decide, should I bind? Should I not bind? If I bind, how long should I bind? Because if you bind longer, there is more signal. If I bind lesser, there is less signal. That is exactly how all systems in our body work. One protein fixing into other system, another protein, and giving them instructions.
Wow. So these systems have, let's again say, a spot which has a very unique shape.
Yes.
And there's only one single protein fold which can take that spot and fit in properly.
Yes.
So it's like a jigsaw puzzle with a million different a million is a very small number, an infinite number of different um, permutations, and there's only one which fits in exactly.
That only one will fit exactly. Nothing else fits.
Wow. So
And
just to make a small analogy here, this is very similar to how authentications works in softwares. So you have these passwords,
okay.
and you want it to create a different hash.
Yeah.
Every time you type in. So basically,
What is a hash?
So hash is it's a conversion of from the string that you type,
okay.
And it is encrypted, and there's a kind of a different string added to it. Let's just call it it's called salt. I don't know why, but so there's the password that you type in. There's a salt, and then it's encrypted. And since the salt can be changed or kept constant, and you have the string, so these two, when they are put together, will always create a unique hash.
So that means, that is why all the passwords are unique.
Yeah, the passwords might be same for the on the human level,
That's right.
but
But
when the hash is created,
yeah.
it's probably going to be unique. And there's on the database, or there's kind of a table for each website or an app, and there's only one password which can fill in that particular which can exactly be equal to that hash. So
Wow.
it just blows up my mind how these two systems are correlated, and maybe the this entire this concept of cybersecurity was inspired from DNA and cells.
I have no idea. I mean, that's really revelating information for me. I didn't know about it, but that's true. I mean, yeah. I mean, it sounds very similar because proteins have this unique structure to bind to this thing, and especially a lot of our medicines which we take work on this principle. So, for example, there is some disease caused by, say, one protein attaching too much on one protein. Okay.
Mhm.
If I want to stop that through a medicine, so what does the medicine do is the medicine has a unique shape which binds to that protein earlier than the other protein could bind. So it's competitive. So if the medicine is binding, that protein cannot bind to the other guy.
Yeah,
So that's why you're kind of like stopping kind of thing. And a lot of the medicines work on this idea, like not allowing this unique jigsaw to lock with each other. And the folding
yeah.
itself is actually very important because if they don't fold properly, it leads to diseases, your Alzheimer's and or your Huntington disease. They are problems because proteins are not folding properly.
That's okay. That's I didn't knew that. So it's really at the basis it comes down to these protein folding and DNA sequencing and mutations.
Yes.
So does every disease or disorder or normal functioning really comes down to the how these cells perform their duty, how precisely they do the task which they have to do.
That's right. That's exactly right.
Well, I think that's yeah, it's kind of it justifies why these diseases, disorders are so tough to uh, cure and so hard to find a cure for these uh,
We have such
diseases.
billion options and we do not know how are they folding. So where
And they are very small.
Yeah, they're very small. They're so you can't look at them. You just can't like give a child some DNA. Oh, you know what? Let's play some experiments, like fold it wherever you want. It doesn't work. And there has been a lot of computational work, or really, a lot of the people are using computers, softwares, programming to predict this kind of folding. It does for small structures, we can get it, but we are still much far away from predicting something big.
That makes sense because there are almost infinite number of states which is possible to create, and even a computer cannot compute that many states.
It's and
That's
that's
really
all,
interesting.
that is all happening in every single cell in our body right now.
Just a lot of work
Yeah, it's a
being
lot of
done in the background.
exactly.
So
So
coming back to the protein folding.
yeah.
So what happens after the protein is folded, and uh,
Okay.
how does it proceed after that?
So after the protein is folded, actually, I mean, it's almost like an Amazon delivery system. I'm telling you why. Because so it's like, okay, now the cell is like a factory, right? It has a heart and everything. It's producing stuff. It's factory. Now,
Yeah,
once you make a product in factory, what do you want to do? You want to send it to the wholesaler, the retailer, the customer. That's how it
right,
works. The cell also wants to send this to different organs of the body, to different other places, and to
right.
work. So what it does is it uses this structure here, as you can see, this guy here called Golgi apparatus. Golgi was the name of a scientist, so it's not an English word. So this guy here, what it does is it packages this protein. It packages into, it adds something, it so that it can move across cells smoothly without any obstruction. It's almost like giving it a card so that the toll and the import tariffs are not charged for the protein.
All right.
So it makes those kind of cards and those kind of authentications and packs this protein and sends them out of the cell. And once they are out of the cell, they enter different ways. It can enter your blood, and the blood helps in supplying all these to different organs of the body and things like that. So that is how the entire then from small cells our organs work, our organ systems work, the tissues work, and our body works. And that's same for every single living organism out there, not just humans.
Pretty fascinating.
It is. See, I've been telling you.
So this is how the normal procedure works in your body. It's happening right now. So
Right now, every single second.
So in a second, how much of this process, how long does it take? Does it take 5 minutes or one day or?
No, these happens all in seconds. All the entire process which you saw, making two DNAs from one DNA and then to RNA to protein. These are happening in seconds.
That's a lot of work.
That's a lot of work. So just imagine, like for example, if I want to make a specific protein, I have to search, like, because proteins are making proteins, as you saw. So
Yeah.
I have to search that specific sequence in a 2-meter long structure with so many, like 3 billion bases, and then copy that, make an RNA, copy that, and make a protein. So that search is extremely fascinating. Like, how does the system does it so quick, so fast? And it's not a standard system. Like, we are working, like we are running, we are crying, we are sleeping, we are hurting ourselves. But that our body doesn't say, oh, I'm hurt, I can't produce anymore. They're still working.
We can all take a page from the book on hard working.
Yeah. But that's exactly how they do. Like, they search for exactly, and they make mistakes. It's not like it's 100%, but there
One
are
in a billion.
1 in a billion, and there are proofreading systems. There are proofreading systems, like, which will go back, check if it is little bit not right. It will cut it out, delete it, replace it. So there are proofreading proteins also available in our system.
That's insane. When you talk, it's so fast and so small, and so this proofreading system is present in every cell.
Every single cell. So for example, if a G is paired with a T, so what happens is then the DNA, the bonding will be a little weak. So it will become like the structure will become different. It's a signal for different proteins to say, oh, there is like a bubble structure. It is something problematic. We need to go there. They check it. Oh, it's not the right thing. Cut it out. And then bring back the other guy, fix it, and then go back.
That's insane.
That's I mean, it's extremely interesting, like small things. For example, when I was talking about how the DNA has to be opened up, so you might think, like, it's natural for us to believe that it will take a lot of energy to open up these two strands. So as I told you, that A and T form only 2 bonds, G and C form 3 bonds. So which is easier to break, 2 or 3?
2.
2 is easier to break. And see, that's the beauty of the nature. Actually, there is more A and T present at the beginning of where you have to open up, so that it's easier for the proteins to open up the DNA. So the design is already made. It's predicted that this has to open. So we should not make it very complex or very strong. We should make it a little weak. So they put more A and AT. So even though it's random, there is a lot of order in that.
All right. So it might be possible it's not random at all. It's just a very sophisticated optimization, which we do not understand.
Exactly the thing. I mean, it looks random because I'm like, we can't find a pattern. It's not like a Mensa puzzle. We can't find a pattern like 2 + 2 is 4. But there is a pattern which has evolved for billions of years since the formation of a planet.
That's right.
So it's a long process, and that's how we end up with small. That's an entire journey. It took 1 billion years probably for that one coronavirus to develop that spike protein and to infect a human cell and cause this devastation today.
That's right. So I understood about, the entire process, how fast it happens. And so how does the coronavirus and the underlying RNA
Yeah.
disrupt this process?
So they are not disrupting, that's the thing. They are, they, this is a part of the process itself. So the virus is what it is doing. It is just doing it for the sake of its own survival. They want to survive. Every single living organism, the idea is to reproduce and go on. So what they're doing is they're infecting the cell. uh, they are trying to make the DNA, make a factory, their own factory, the virus factory, and trying to infect the other cells around them. But
Right.
our body kind of knows that there can be foreign invaders all the time. We are in fact being attacked by viruses every single second, but we are not getting sick. We don't get infected. Sorry, because we have an extremely powerful immune system. We have these
Yeah.
white blood cells which like sense it. Oh, this guy is foreign, let's go and kill it. And they're doing that structure all the time. So now it depends, because even for the immune system to develop immunity for the virus, it's not easy. It's that these processes, especially for 8 billion or 7 billion of us on this planet, it takes little time for it to become normal. But the virus is spreading faster.
Right.
So if I have to just think from the time frame only, not from life and death point of view, if you
Yeah.
wait for enough time, our body will develop an immune system for the virus.
Right.
But just because we cannot wait for that to happen, we have to like stop the virus, develop vaccine and all. Because what happens in a vaccine, what you do is you put a very small amount of virus inside a dead or a very small amount in a living condition in your own body. You're actually infecting that so that the immune system triggers and they realize that, oh, this is something we have to develop against. So when the real virus attacks you, you are already immune against it. That's the entire system of vaccination.
Right. So there's kind of a database which they check. Okay, we know this is not a good guy,
Yeah.
so we should like defend ourselves from this.
Yeah,
And since coronavirus is new, we do not have an entry of it in our database
that's
yet.
true. That's true. And it's almost like, like, because so many people have seen Game of Thrones. The character Arya Stark had a hit list of people she wanted to murder. And that's, I think our immune system also maintains some kind of hit list for people they don't want. And that's how it works. It actually, they have to develop a system to fight it out. It will take time. And this the bit of a scary thing is, because viruses are there, millions of them in the air everywhere, and new viruses always keep on coming out. And especially with coronavirus, because a lot of it has been discussed about how it originated. And we know that it has some animal source, and we have to understand that we did not interact with animals that much. Interacting with bats and all was not, it's not part of the way we were designed to be. The more we are cutting forest and entering deep into the nature and trying to interact with these people, these animals who are losing their lives and their their homes. So the viruses which are present in them, they are immune against them. That's evolution. But we are not.
Right.
So they interact with us, and we are not prepared for that. And that's because we should not have gone there where we have gone.
Makes sense.
And it's the same thing even with the, because there have been also been talks about the glaciers melting, because the glaciers also have a lot of viruses stored in them. And and they're frozen like for millions of years. So that means we don't have immunity against them. So once the global warming takes more and more into the entire environment and more glaciers melt, if these viruses are released in the environment, we really don't know what is going to happen. So we have to always take care that our actions can really lead to such kind of catastrophes in the future.
So this somehow even relates how climate change can also lead to a next virus outbreak.
It does, climate change. And that's what scientists are actually saying, that if we are not really working on global warming, probably the next virus attack, the pandemic might be from a climate change issue. I mean, this is also predicts climate change, but from glaciers and all, that could be the next big outbreak.
I also read an article which said uh, coronavirus and other such viruses are already present in animals in the Amazon.
Okay.
And you can probably predict what other virus outbreaks can happen if you study what viruses are present in that particular forest. Is that true? And
Well,
like,
it's a difficult answer, but in just from if it's true or not, yes, you can predict a lot from just studying organisms. And as in fact, even this coronavirus, not this exact sequence, but that from bats we will have another kind of virus outbreak was predicted somewhere back in 2015 itself. It was published in Nature magazine, and it was there. We knew it. So of course, we did not know this exact thing would happen. But the thing is, like, again, it comes back because the scientific journal is very distant from the public administration. Scientists are not making policies out there. So even if you have an information in journal, it stays in science. But to tell it to people, oh, we may have this, so let's try to develop a vaccine even before the outbreak has happened. That type of proactiveness we still don't have, but it would be required, I mean, given the situation now. But we might not be able to predict exact outbreaks in the future, but we can at least predict something similar, like this may happen. And even if we develop vaccines or something for that, even if something similar kind happens, we may already have a solution. So that's like a learning thing I think we should take from the coronavirus. Sorry, Josh, your audio is muted.
yeah,
Sorry.
I'm sorry.
Yeah.
Yeah, so that's something we should definitely, take away from this and something to think about even after this whole pandemic is over.
That's true. That is something we have to think over. Like, how do we prevent the next big outbreak? And it would require a lot of work between the scientific community, the administration, and because uh, identifying a virus, a structure, what we talked about initially, is one of the first few steps we are doing. But from that to medicine is a long process. It's not fast. Sometimes it takes 10 years from a lab to a market. So
Yeah,
that process is a very long one, and we have to be proactive about it, identifying viruses and trying to develop vaccines and strategies for it beforehand. That's the only way I think that if there is a next outbreak, we can manage it much better, and we don't have to go down this global lockdown for so many months.
that's true. Well, it was really great. I just got a so much better perspective on not just coronavirus, but my own body and how things work, and it's really great. Yeah, I didn't knew biology can be so much fun.
Biology is fun. I've been telling you for so many years, Josh. But I'm telling you because we did touch upon the computation and robotics, because I do believe personally that a lot can be done with computers now. We didn't have it because DNA was discovered in 1953, and we didn't have computational that time. But I think a lot can be done. So the future is exciting.
That's true.
Yeah. But it was great.
And thanks a lot. And for everyone who's watching here, so Trishit has started a blog and a video channel on YouTube, which is called Emulsions. And the goal is really simple: to help people like me and everyone out there who doesn't know much about some particular concept and who doesn't really understand the jargons of that particular field or domain, but wants to know more and can't read the science papers and understand better. So he's really helping out by condensing that information in a very, very nice and understanding, simple language. And he's doing that with in the blog format and on YouTube videos as well. So I'm really, really excited for what's going to come up. And I think there should definitely be one video on DNA as well. I think it really needs one video segment of its own.
Yeah, maybe I should think over it, but it's fun, right? I mean, because
Yeah,
I try to give
learning is fun.
a very small little way, but it is the more you try digging into it, I mean, it's extremely, the research is, some of the research papers, how exactly that unfolding of DNA happens. You have one papers. It's too detailed.
That's true. The protein folding and the DNA sequences is something which really which is really fascinating for me.
Oh, that's great.
Also, how quickly it happens. I would definitely like to know more about it, and maybe reading those papers will be a bit easier for me now.
It would be, and I'm sure, because it's going to be much easier if you read more. If you read now technical aspects, if you read technical articles on these sciences, on these processes, I'm assuming it will become a bit easier for you now to understand that what's exactly happening.
If you can really, put some links to some of the papers on the stuff which we just talked about, that would be really awesome for me and all the viewers.
That's actually a great thing. So maybe, yeah. So I'll just select a few papers, and I will also add them when I'm posting this video, so that everybody can refer to them. And if they have questions, they can always come back to me.
That's great.
Thank you so much, Jess. It was great talking to you, and you'll always
It was
come back to
fun.
this place for
Thanks
everything.
for having me.
No, not at all. Thank you so much.
Yeah.